Hey, how's it going, class? It's Mr. Miles coming to the podcast. In today's podcast, we are going to be reviewing bacteria. Alright, now if you remember, first thing we talked about was viruses. Now, viruses sit outside of the realm of the living because they don't fit the 8 characteristics. Yes, they do have genetic code. Yes, they can reproduce, but in all how they do that, it's a little bit different. They don't have cells. They also can't react to stimuli. I'm trying to think of some other things. I don't know. It's pretty late. I'm sure you guys can come up with plenty more. Oh, homeostasis. They're not going to be able to do homeostasis. And there's plenty of other things that keeps them out of the realm of the living. However, if we go inside of living, you go through the 8 levels of classification. Remember, that's the 8 levels of classification, not 8 things that make something living, but the levels of classification. You have domain, kingdom, phylum, class, order, family, genus, and then species. In our domain, we have 3 groups, and the first one being bacteria. Now, along with that is our archaea. Now, the archaea and the bacteria, these guys are both known as the prokaryotes. Now, originally, it used to be you had the prokaryotes and then you had the eukaryotes. And so all bacteria were just simply known as prokaryotes. That was their Latin name. But then along came the archaea. And remember, these guys live in extreme environments. So it's going to be very hot. It's going to be very cold. Very acidic, very dry. They can live in places that we would not expect life to be. That's really the only difference between archaea and bacteria. So that's why they're both prokaryotes, but we are nice enough to give them 3 separate domains. Now, ignore eukaryotes for right now, because this is what we're going to get into in the next podcast and lessons and stuff like that. Right now, we're just talking about these 2 guys, bacteria and archaea, and the domain. Now, remember, what makes them unique is that they're both unicellular, and they also have no nucleus. Remember, nucleus is that brain part of a cell. So there's the brain, and it's going to contain the genetic information of, in many cases, DNA. Sometimes, if you're a much simpler organism, just RNA. So these 2 guys, bacteria and archaea, are very similar, except for the extremophiles thing, so extreme environments. But they're going to match a lot of these. So as I go through bacteria, just understand that a lot of this does apply to archaea. They're very much like it. Now, speaking of genetic information not having a nucleus, because they don't have a nucleus, they have a very simple structure. So there's this. So they will have a flagella, or flagellum, however you want to say it. So they will have a flagella. They will have, some of them will have a cell wall, but all of them will have the cell membrane. Okay, and remember, it's that semi-permeable membrane that lets certain things in and out, and other things not so much. Wall is going to give it that rigid structure. Not all of them will have that. They will have ribosomes, dotting the inside, because they need to be able to make proteins just the same as anyone else, because remember, ribosomes makes proteins. And then instead of having that nucleus, they will just have their genetic information is kind of jumbled up inside of there. Okay, so in some cases DNA, sometimes you just get simple RNA. It's not as cool as ours. It doesn't do the double helix thing. It just is a single strand instead of like a double helix that wraps around itself. It does something like this. Okay, so that would be, if you're looking at the DNA inside of a human, theirs it's just straight line right down. Doesn't match either side. Now, that's the simple structure of it, but understand that these guys are not simple. You have plenty of different types. You have ones that are spiral shaped, and I can't draw spiral shaped in 3D to save my butt. But they look like, I don't know, almost like spaghetti noodles. You have ones that are rod shaped. You have all different sorts. You have globular shaped ones. But bacteria come in a variety of shapes and sizes, and they're all magical and wonderful in their own right. Actually, most bacteria, even though we think of them as being bad, most of it does absolutely nothing for us, whereas other ones are very good for us. But then you deal with the ones like E. coli. And even with E. coli, it's only certain types of E. coli are bad for you. Some are actually plenty helpful for you. But it's just when you get the nasty strain. You get streptococcus, which causes strep throat, which is having a lot of these guys growing in the back of your throat, causes inflammation, gets you very, very sick. So, yeah, and they don't all have to have flagella. Again, some of these you can see that they really wouldn't have flagella. They just do it like that. Now, how do we get more of them to the point where you actually could get sick from them if they are bad? Remember, there's 2 different types of sexual reproduction: asexual and sexual. Now, in asexual, they're going to undergo something called binary fission. And in binary fission, you are simply going to get one. It's going to divide into two. Is that like the apple booty diagrams? It looks like an apple. Then it looks more like a flat booty, kind of like Miley Cyrus's. And then boom, you get two different ones. They're going to be genetically identical because obviously they're just coming from the exact same parent. It's only one parent in this case because it's asexual. Now for them, they're going to do sexual-ish, and I put ish on there because they don't actually. You take two bacteria, and normally what would happen is when a mommy bacteria and a daddy bacteria love each other very much, they get married and they have a baby bacteria. That's not what happens. Instead, what's happening is they will simply exchange genetic information, so that way they can get variation. So there may be something in this genetic code that this one picks up, passes on to that one, and now you have two completely different bacteria than what you start off with. So that is sexual, because you're getting that variation. But because you're not producing offspring, we add ish to it. But of course, when you get that variation, then you can do binary fission, and now you have a whole bunch of new types of bacteria. And so what this is called is conjugation. Okay, now we got to be wary, because what happens if these guys are bad? How do we take care of them? How do we, they're reproducing and our food supply on us, on the ground. Oh my gosh, we're going to die. No, there's ways to combat it. You can use antibiotics. Now, in the case of antibiotics, this is just going to be medicine that can be administered that kills bacteria. Of course, the most famous of all of the antibiotics is going to be penicillin, coming from the fungus. So it's actually coming from a fungus, Penicillium, that was actually discovered and turned out to be able to kill bacteria. However, bacteria do have the ability to become resistant to antibiotics. Just think of it this way. Let's say you have a whole bunch of bacteria on here, and all of a sudden, a new antibiotic's brought in, and it wipes out, I get to use my little eraser, and it wipes out all the bacteria except for one. Now, all you'll have is one single bacteria, but he is resistant to antibiotics, so he is free to asexually reproduce and can make more of them, and then you simply have to find another antibiotic or you're going to die. And so we constantly have to change our antibiotics to match the mutating, conjugating bacteria that are changing all the time. Now there's a very easy way to do it if it's not something that we care about killing, or you can't really kill. So you can't do this to a human, but you can do it to milk. It's called pasteurization, and obviously we can't do it to humans because it involves extreme heat. Remember, we're talking about bacteria. Archaea, they're not lethal because they don't exist in our environments, because they need extreme environments. Bacteria, however, don't like those extreme environments, one of those environments being heat. So what they'll do to milk is, when they get the milk from a cow, and remember, cows are dirty, and farms can be dirty and covered in bacteria, and milk sits there for a while. It's not like it just shows up at your breakfast table immediately. So there's a lot of chances for bacteria to get inside of there. So what they do is they flash heat it. So they'll put in these big vats, they'll crank up the temperature really, really, really high, but then they bring it down fast enough to where it doesn't really change any of the molecular structure of the milk at all. It doesn't change any of it. Don't let any hippies tell you that it does, because it doesn't. Oh my freaking goodness. It's still milk. The proteins are still exactly the same, and you can use the internet, and as long as you're using credible sources, you'll find that that is correct. You can flash heat milk through the process of pasteurization. You can kill any bad bacteria in there. So always, always, always drink pasteurized milk. Oh my gosh, it's silly not to, because unless you really feel like you're so cool, milk that hasn't been treated for bacteria, and you'll get really cool because you'll die and your body cools down when you get cool. That's where the joke was going. Anyway, so we have different ways of killing bacteria. However, we don't want to kill all bacteria. Some bacteria is our friend in the form of decomposers. This is probably one of the most useful things they are into the environment, is decomposers. So that means they're going to eat dead or decaying things. It's mostly they're going to help with the decay process, because you go, oh well, my turkey sandwich is not alive. Yeah, I know, but stop being a smart aleck. So an animal dies in the woods, a tree dies, any organic material dies, and bacteria helps to break it up. Now there are funguses that do the same thing. There are some insects that will do it, but bacteria are our main guys. That's why they're the reasons why things smell when they die, because as soon as they die, bacteria starts growing, and you're smelling that delicious bacteria breaking down it into very simple molecules that can either get airborne or you're smelling the residue of the bacteria themselves. So they are very important for the environment in the form of decomposers. Other really cool things for bacteria is, let's say you have bacteria and bacteria is sad because all of a sudden it gets very dry. Oh no, this sucks. Bacteria will revert into a stage called an endospore. And in the stage of an endospore, they can survive for a very, very long time until wetness comes back. So when the wet comes back, then the bacteria will simply come out of the endospore. Now, what's in the endospore during this time period is it simply just goes to sleep. So they're just sleeping, and it builds a nice hard shell, which almost looks like a beard afro. Looks like my dude's like Amish. But yeah, my little Amish bacteria has this little hard shell, and then when it becomes wet again, he sheds his hard shell. Apparently, gets punched in the eye and has a black eye for some reason. It's really creepy looking. But anyway, that's what an endospore is. So that's the wild, wacky world of bacteria. So again, not a whole lot of information to remember, but for your small little fragile brains, it just might be. So make sure you understand this, because if you don't, then you might get an F on the test, and that would not be any good. So don't do that. Alright, cool. I'll talk to you guys next time. Alright, bye.